today's lecture will be slightly interactive if possible, but don't worry if you don't know any of the answers. I spent most of my second year not really knowing what was going on, and it turned out fine. So, high cerebral functions. This doesn't come up much on Oxford MCQ questions. There's about 5 questions in the entire question bank on them. But what it does come up in a lot is, firstly, as an individual essay topic that not many people do or do well, and therefore if you do an all right essay you tend to score much higher marks than, let's say, if you answered a visual essay. And the second one is it comes up a lot that you can kind of add it into other essays. So it's a really great topic that you can kind of translate across, especially if you are doing an essay where you're not really sure what to write, or if it's a more conceptual essay like about plasticity. So the first half of this lecture will be about sleep and the second half about memory, because they're both kind of tied in. So today we're going to learn why we sleep and also have a brief overview about the concepts and theories about how sleep happens and how we wake. Okay, so does anyone know why we sleep? Okay, that's a good answer because no one knows why we sleep. But we know that there are different types of sleep. So stage 1 is generally when you kind of are asleep but awake and you keep on waking up at night, but you remember that you're dreaming. That's kind of stage 1. Stage 2, 3 and REM, much deeper sleeps, and that's kind of what you want to achieve at night. So this is very much part A stuff. So if you just memorize what features are in stage 1, 2, 3 and REM, you'll be absolutely fine. And this is how we measure sleep. So polysomnography is kind of on its way out. As you can see, it's very involved. It looks like a jellyfish on top of her head. Whereas the actigraphy, which you can just basically use, is basically like a Fitbit. And it's being used so much, especially in sleep studies in psychiatry, because in many psychological illnesses, you have disrupted or changed sleep. And here it's a very non-invasive way that people can go about their lives, and we can still record the data. So why do we sleep? Well, firstly, one theory is that we need to sleep because our brains are constantly having neurochemicals, neurotransmitters fired at them by other neurons, and when we sleep there's less activity, therefore less neurotransmitters, therefore there's no adaptation or resistance to the neurotransmitters created. However, this is a very flawed theory because, as you know, we keep breathing, etc. during sleep, although alters and rates, etc. But these neurons, neurotransmitters, and also some pathways actually increase in activity when we sleep, and these pathways also don't become resistant. This is a really nice study that you can kind of critique on quite a, common sense level. Okay, so we do know that sleep's really essential though. So Randy Gardner is my favourite guy to bring up about sleep. He's a high school student who decided that I'm just going to do for my science school project fair, I'm going to just stay awake and see how long you can stay awake for. So eventually a Stanford professor became involved because they heard that this kid was staying awake for over 250 hours. And then the Air Force decided to get involved because sleep and wake is very dominated by the aeroplane industry, because obviously you want your pilots to stay awake as much as you can. That's safe because that's economically beneficial, but also in the Air Force because you want your fighter pilots, etc., to stay awake and be safe and effective for as long as possible. So basically Randy stayed awake for, as it says, 264.4 hours, which is quite a long time, and by the end of it he was very, very strange. He had huge cognitive deficit, so he couldn't remember between about half a minute when you were taking away 7 from 100. So the 7 from 100 test is a standard bedside test for delirium in hospitals, so it's a nice clinical element to add. So you ask the patient, can you take 7 from 100 and keep on going? So you go 100, 93, 86, 79, etc. But he forgot what he was doing by the time you got to 65. That's how sleepy he was. But it's really interesting to see that he bounced back basically in terms of scoring normal on cognitive and behavioural tests after only 14 hours and 40 minutes. So we can survive without sleep. We don't survive optimally without sleep. and we can also recover quickly if we don't have sleep. Okay, so sleep is also important for motor skills, not just cognitive tasks. So as you know, when you're sleepy, you don't want to move that fast, you're not going to perform well in activities. And here's a really nice study that basically summarizes that everything in terms of sport decreases when you haven't had enough sleep. Okay, so it's a global phenomenon of sleep deprivation. So sleep, as I mentioned before, is altered in a lot of diseases. So in bipolar disease, when patients are manic, they feel that they have no need for sleep, okay? Whereas in depression, which is basically the opposite of mania, patients often stay in bed a lot, they sleep a lot, they're hypersomniac, okay? Very interestingly, in delirium, which arguably is something that Randy Gardner experienced, delirium is characterized by reversal of sleep and wake. So that means that you sleep a lot during the day and you become awake at night. You basically become nocturnal. The mechanisms of this is not elucidated. However, we know that in delirium, acetylcholine and dopamine levels are severely skewed, and so that might play a major role in why these cycles are flipped. Okay, so as I said, important circuits include acetylcholine and dopamine. However, there's a load of circuits and neurotransmitters involved in sleep and wake. So I really want to get across this concept to you about sleep. It's a global brain activity. Your whole brain changes, your whole body changes when you're asleep. That means it's going to involve a lot of circuits, okay? Every single circuit in your brain is basically modulated when you sleep to induce the sleepiness and to maintain it, okay? So it's not just going to be one circuit that's affected and drives this, especially as you can see, sleep is very important, as you can see with Randy, but we haven't actually fully understood the dominant circuits in terms of, is acetylcholine more important than dopamine? Is it more important than orexin, etc., etc.? Okay? So we've seen the global level, lots of circuits going on, lots of targets, lots of neurotransmitters. I'm going to talk specifically only about orexin, which is also known as hypocretin, because you can tie it really nicely into a known clinical syndrome, which is quite rare for sleep because not that much is known about sleep. So orexin is very important. That's all you basically need to know. It keeps you awake, okay? Without orexin, you fall asleep. This has been shown in rats, firstly, by targeted neuropharmacological action via agonists of orexin 2 receptor, but it's also been shown in knockout rats since, okay? So lots of murine mice. However, we know its importance in humans because of narcolepsy. So does anyone know what narcolepsy is? Okay, I'm going to keep on talking. I hope you guys are still there. It would be helpful if someone said that you can hear me, just so that I know I haven't been talking to myself.
Grace says, keep falling asleep.
Yes, exactly. Okay, thank you. You do keep falling asleep. Another really important symptom that people always forget about narcolepsy, and you can kind of remember it by thinking that they fall asleep easily and they go weak at the knees with laughter, and that's cataplexy. So any strong emotions, it's basically as if a puppet's strings have been dropped. They lose muscle tone, they lose control, they become temporarily atonic and paralyzed, and they fall to the ground. So especially when they're laughing, they become weak at the knees and fall over. So why does orexin do this? Well, firstly, you're obviously going to fall asleep if the things that really contribute to keeping you awake are not there. But the cataplexy is really important because it ties in emotion to sleep, it ties in motor control to sleep, and it also ties in general awareness to sleep, okay? Because whilst people are having cataplexic attacks, they are aware that they're having it, okay? And they sometimes know before it happens, they have a chance to sit down or grab onto the side of a rail before they fall down onto the floor. So cataplexy really demonstrates the interlocking between emotion and sleep. So you can also see this through maybe depression or mania, and it also ties it through to arousal. So just because you're sleepy doesn't mean that you lose your awareness. Okay, so I just want to have a look down here about the treatments for narcolepsy. No treatment is actually very effective for narcolepsy, and often it's kind of more of a management of symptoms and quality of life than treatment. We haven't actually found a replacement for orexin neurons. We haven't found agonists for remaining receptors, and we basically hype them up, these patients, so that they stay awake for as long as they can before they have to have a nap, which unfortunately is inevitable. So we've talked briefly about how to stay awake. We've talked briefly about how to stay asleep, but you need to consider that to fall asleep and to wake up are very distinct processes from maintaining sleep or wake. It's a transition, so it's the turning off of some systems, it's the turning on of some other systems, okay? So anatomically, the ascending reticular activating system is key in this transition process. It's not that much known about it, but they do know that in diseases it changes, and also, and it's specifically spared in locked-in syndrome. So locked-in syndrome are when patients can't move their entire body except for their eyes, and they're essentially in a coma. However, they're not comatose because their arousal, their awaking system, is still on, so they can still control their sleep-wake cycle. So moving on from the ARAS, here again we have this global concept, as you can see in the top right corner figure again. So it's not just one network that says awake, asleep, okay? It's a seesaw, and it's the net effect of the different activity of all the different circuits. Okay, so it's not a second that you just fall asleep. It is a process, and you yourself probably all know this. When you lie down, you have to close your eyes, you relax, you pretend to sleep for a while before you fall asleep, okay? But once you do fall asleep, there is a moment where you're conscious and unconscious, but it's a very slow point to then get to the transition stage. Another theory about sleep-wake transitions, which isn't contradictory to this seesaw model, is that you build up a debt of sleep. So whilst you're awake, you get more tired throughout the day. So either the balance between these circuits start to tilt, and that's why you feel more sleepy and you have that sleep debt, or there could be some molecule that builds up in your system and then at peak threshold meets a threshold that then causes you to fall asleep. Okay, so this whole sleep debt cycle, I want to make very clear, this model is not at odds with SAPER 2010, but it instead can be translated to it. However, most often this sleep-wake cycle model is used in terms of adenosine. So adenosine, as you probably know better than me, as you're in preclinical still, it builds up whilst you're awake. So this is one of these molecules that is also linked to energy, okay? So it makes sense that people have been looking at adenosine as a candidate molecule to control the sleep-wake cycle. But the argument basically is that once you wake up, adenosine starts to be released, and more of it builds up throughout your body until you fall asleep. And then as you fall asleep, during this sleeping time, it degrades naturally, metabolically, or it's reabsorbed, and then the whole process starts again. Okay, so this sleep-wake cycle could be due to the seesaw effect, or it could be due to the adenosine. Right, does anyone have any questions before we move on to memory? Let me exit and see if I can see the chat. No, oh, Grace Wheeler is typing, this is exciting. No, that was all good. Okay, thank you, Grace Wheeler. I'm going to go back to my slides and you can see them. Can someone speak, because I can't see the chat?
Yeah, we can see them.
Yeah, we can see.
Thank you very much. Okay, so memory. Right, so firstly, you need to consider why memory is talked about a lot hand in hand with sleep. So you probably know that when you sleep more, you remember better, right? When you're lack of sleep, you forget things, you're a bit more grumpy, you're emotionally not ready to learn things. So there's also a reasoning and a model that says sleep enables memory because it allows time for the brain to not have to like deal with daily life and daily stimulus of, oh, there's a light, oh, there's a car, oh, I need to cross the road now, oh, I'm hungry. Instead, it allows the brain to have a few moments to consolidate the learning and to consolidate the neurons. Okay, so I want you to have that in your mind as we go through memory, because it often comes up. You can say a lot about sleep and memory essays, and that's when you're going to score really high marks, because it shows that you actually understand the concepts between plasticity, sleep and memory. Okay, so as you know, there are different types of memory. This is very basic stuff. It's just one of those things you have to memorize. I'm very sorry about that. And you've probably been well too aware of the boxes, water mazes and conditioning. Yeah, so I'm not going to go through those because you can read about them in Kandal et al. And it's a great book, a great section on water mazes, but it's much overemphasized in Oxford Medical School. It shouldn't really take that much part of your essay, maybe 1 or 2 paragraphs if you're going to write an essay on it. Instead, you should use this and then translate it to the circuits and mechanisms. So memory, again, I'm going to beat the drum about a global function. So a lot of things are involved in memory. The oldest thing is smell and taste and vomiting. So a ancient mechanism for survival was that you would learn very clearly that if you ate something that then made you vomit, you would never eat it again. And the olfactory center was involved, the amygdala, hypothalamus, hippocampus, and the mammillary bodies. And so this mechanism is one of the oldest examples of learning. So it keeps us alive by stopping eating us poisonous things. Okay. Another part of the brain that we now think is very key in terms of modern day memory functions is the temporal lobe. So you all know about patient HM. Please make sure that you know what actually was taken out of his brain, unfortunately, when he had his treatment, because many people forget. They say it's only the temporal lobe, but it's actually a lot of other things were taken out, which are also very key in memory. So he was able, though, to still do crosswords if he adapted existing memories and kind of absorbed new information. Specifically, he did a crossword and everyone was amazed that he would answer something about the new development about polio, but he managed to add it on to an existing memory about polio that he already knew. And that was the only way he could add memories. And he could only do a few of these because it took a huge amount of rewiring of his brain to do it. So another way to remember at the temporal lobe, oh, I think I froze it. Oh no, there we go, is the Kluver-Bucy syndrome. Okay, I love this syndrome because it's very easy to remember. So temporal lobe and amygdala, and there's a weird amount of symptoms that come along. Okay, so they like to lick things. They're basically, you know, like dogs explore things with their tongues. That's how these people then explore the world. Bulimia, memory disorders, flattened emotions, inability to recognize objects or faces, or also known as prosopagnosia. Sorry, I can't say that word. I'll type it in the chat later. It's just like the man who mistook his wife for a hat, if you read the Oliver Sacks books. So you can see again the temporal lobe, although it functions in memory, very key, it also functions a lot of other things. Again, the very interesting link between sickness and vomiting with the bulimic aspect of this temporal lobe and memory. Another clinical syndrome that you can write about is Korsakoff syndrome. So does anyone know what causes I've written it down. Thiamine deficiency and usually alcoholism. So I remember Korsakoffs as a brand of pickles because these people essentially pickle their brain in alcohol, which essentially is what happens. Basically, they literally pickle their brain. So a huge amount of demyelination brought about by lactic acid action due to thiamine deficiency, but it is reversible. Okay, there's a precursor. It's a triad of symptoms called Wernicke's encephalopathy, and that can be treated if recognized with very simple cocktail of vitamins. So vitamin B and C are generally given, called Pabrinex, that's the brand, to treat Wernicke's encephalopathy. So it's really sad when you see Korsakoff's patients, because you know it's completely reversible. They're often very young people, and there's also a growing number of them being in kind of the 20s to 50s young female population, which is quite interesting in terms of a public health point of view. So how can you use these in your essays? A memory and sleep essay, or just a memory essay, you do your introduction, you define the types of memory, You then talk about the experimental basis. You go for water mazes, boxes, Pavlov's dog, etc. And then you want to take it to a human level. So what's the basis of our understanding of memory in humans? Well, it's when memory goes wrong. So you can talk about Alzheimer's, Korsakoff's, you can talk about Clover-Bussy syndrome, you can talk about patient HM. Okay, there we go. And then you move on to how memory is a function of plasticity, the mechanisms behind memory. And this is where you can translate it to sleep, to wakefulness, and to learning, because memory and learning go hand in hand. So just like if you can think about something, you know, short-term memory cramming, for example, for an exam, this is what happens when your neurons temporarily make a connection. It doesn't last that long, and they move on. So it's like you know, speed dating. But long-term memory, you can think of it as a very long-term commitment. They've formed a relationship and they're staying. So they've had that initial kind of sniff around each other, the temporary ooh, and then they've decided, yes, I'm going to make a proper connection, and that's how long-term memory is made. So historically, we kind of thought that neurons did this. They sniffed around, they made connections, and you know, it's a constant process of forming new connections. However, it was fully found, and this is Hebbian's theory of learning, to be equal to synaptic plasticity with these two complementary theories. Okay, so these are the two ones you really want to learn and focus on. The fact that two neurons that are there at the same time, you know, they're willing to date each other at the same time. There's some activity, some sparks fly, and then eventually activity is reciprocated when someone, you know, one of the two in the relationship says something. Okay, sorry, it's quite a bad metaphor, but this is how I always remember how memory works. And then the Kornosky theory is quite interesting, because it implies that previously temporally related neurons can form strong relations, not necessarily of their original function, but also implying that these relations aren't just activity based, but they're also changes in the actual physical properties of these neurons, which have since been proved too. So when two neurons meet, when you learn, there's activity changes, it changes the physical properties, and these changes can be temporary or they can be permanent, and that represents short and long-term memory. So looking mechanistically, so we've looked huge globally, we've looked at the experimental view, we've looked at the human view in terms of clinical presentations, we've then looked at conceptually what we think is going on, and now looking microscopically at calcium, because I know preclinical examiners love calcium. Basically, they think the relationship between these two neurons are done by two things, calcium and NMDA. So NMDA and calcium are linked as well. So at the postsynaptic neuron, the MDMA is blocked by a magnesium ion. So calcium can't go through. And basically, when both of the pre and postsynaptic membrane are activated, just like we've seen in the Hebbian and Kornosky theory, then the magnesium leaves, calcium is allowed to enter, and then the neuron learns, basically, and becomes permanently active about when the presynaptic and postsynaptic neurons are active, synchronizing activity, and therefore reinforcing learning. So briefly, the voltage gate calcium channel is also very important, because here we're not sure whether you also need concurrent NMDA LTP to happen, or if it can happen on its own. It's basically the same theory, the same basis, and so we know that learning, human, neurons meet each other, form a relationship, and calcium is the key. Okay, So why does calcium make this change? Looking into even more detail. Well, basically, we're not really sure. Here I've got some papers and some evidence, but what we do know is that the CAMK2 definitely mediates it, except for in postnatal development. And I really want you to think about this. I know it's hard to put your clinical hats on sometimes, but postnatal development is one of the most accelerated development stages of your lives. Okay, you've come out from your nice amniotic fluid bath, and then suddenly, hello world, and you're learning a huge amount of information. It's your first interactions with people. You learn how to make motor movements. You learn how to breathe on your own. You learn how to control your emotions. You learn how to communicate. Okay, so it's very important that you understand that the CAM-MK2 is low postnatally, because it indicates that firstly, LTP does not require it. Even though we think that it's a major mediator, it doesn't require it. Secondly, there's got to be other mechanisms for LTP. And thirdly, does this point to postnatal memory and learning being a different mechanism and explaining maybe its accelerated process compared to later life learning? Who knows? Okay, so the Kornowski method also said that there were changes in the actual physical neurons, and here you can see how calcium can also lead to this. So if you take away one thing from this lecture, just think memory, calcium undermines everything basically. So we've shown that the actin mediates cytoskeletal arrangements that lead to these new synaptic structures. So it enables that physical bond between the neurons and makes sure that they can actually go sniff out other neurons if they want. Okay. Other changes, loads of other changes. This is a great slide, great figure if you can remember it, if you want to write essays, because here it basically explains everything we know about what happens physically to neurons when learning and memory takes place. So another interesting thing about the spinal morphology is that using Golgi impregnation and 2-photon electron microscopy, there's an increase in spine number of these neurons. Okay, so there's increased neuron density when you learn, especially in the hippocampus, the cerebellum, and the amygdala and the olfactory cortex. So it's not increasing the spine number, but it's increasing the neuron number, but it's increasing the spine number. So different legs are being grown from a single neuron. I don't know if you can see my picture, but I was trying to show the growth. I completely forgot what I was saying. Oh yeah, the spine number increases, but also there's increased amount of expression of receptors, there's increased neurotransmitters. So basically, once you learn something, once that process occurs, then it's only strengthened in LTP if it's, you know, consolidated. Okay, that's why you can remember some things for life, like how to ride a bicycle. Okay, so here is another example of how the spine physically changes in LTP. And here's a really good review that you can read about the mechanisms of memory in relation to neuronal plasticity. So I think I might briefly skip over this. I'm not sure what time it is. Oh, I'm running quite fast actually. So there are different types of LTP. Okay, so we've talked a lot about generalized LTP and the mechanisms of calcium, etc., etc. But here I've lifted this from Candel. You can read about it in more detail there. But there's an increased probability of transmitter release and therefore potentially recruitment of other neurons or previously silent release sites in this LTP. So this will strengthen the communication between neurons rather than increase it by physical proximity and then increasing the spines. Does anyone have any questions about the mechanisms of LTP? I'm not sure if I'm going too fast or too slow, so do let me know. Okay, I will take silence as golden then, from the current slide. Perfect. So with LTP comes LTD. So LTD is often ignored quite a lot by essays and also just generally by lots of undergrads, which is understanding because it's less boring. Once you get through LTP, you're like, okay, I'm done. I'm just going to write my conclusion and be done with this. But LTP is really important because it prevents a ceiling effect. So if we were currently learning from everything, which we are, because we're currently being stimulated from looking at objects, from moving, from learning how to dance a new dance routine, we're always learning. So if we're always learning, then we're always undergoing LTP, which means that eventually our brains will be firing a huge amount. There'll be so many connections, it wouldn't actually work, okay? So we need to remember the important things, we need to forget the unimportant things, and sometimes we can remember less important things through other memories. So for example, you can think about your favorite memory of your childhood, and then you remember the taste of the ice cream that you had as a child, etc., etc. And that's because you have linked the strong memory of your emotional time as a child. And then if you're eating ice cream at the same time, by chance, oh, you open up that, there we go, we remember. But it's not active memory, okay? So LTD, you can think of it, yes, as depotentiation. Another likening is that in touch, we zone out a lot of things. So it's not useful for us to feel the sensations of our clothes on us at all times, okay? It's not useful for us to see every single light molecule that we, detect every single light molecule that we can detect on our retina. So there's a level of exclusion that allows us to focus on the important stimuli. So it's kind of the same concept, but instead of excluding it at the point of contact, for example, like, I'm just gonna dim down the fact that I'm wearing a jumper on my every single touch receptor on my skin. Instead, it's exclusion after we've absorbed it and after we've learnt it. So here is where sleep comes in again. So some people think that this happens when we sleep a lot, but it's important to know that this can happen throughout the entire day, okay? We don't have that debt of knowledge that builds up, and then gets released every single night, although some people think that this is true. And some people can think that this huge amount of excitation that happens during the day in our brain is the molecule that builds up and then causes us to sleep because our brain gets too active. But this theory has since been completely refuted by ECG monitoring, so monitoring of electrical activity of the brain, and so it's been excluded. But it's nice to mention, and it helps you understand what LTD actually is, I think. So I think I've explained LTD quite nicely, but do interrupt me if I have any questions. So moving on to the mechanism of LTD, well, no one really knows how it works, but some experiments have shown by Bear, Bear also wrote your textbook, that if you have a low threshold stimulation for a long time, it's long enough that you kind of keep remembering it, but if it doesn't meet that threshold, then it will be excluded, okay? Spike timing dependent plasticity is quite a complex concept, and it's not covered by your course, but I think it's important for you to be aware of it in the sense that it's both pre and post synaptic activation that can cause LTP and LTD. It's not just the sending of a neurotransmitter across the synaptic cleft to then bind to its receptor on the post synaptic bouton. Instead, it depends about the calcium spikes on the pre and post synaptic neuron. So you want that simultaneous action, okay? Or that action that's kind of around the same time on a millisecond scale. So again, if you go back to that silly relationship metaphor, it matters that both parties are looking to date someone at the same time, okay? So it really matters about the activation of both parties, not just the postsynaptic party, okay? Really interestingly, it's cannabinoid receptor dependent. So in a second, we're going to talk about drugs in the context of sleep, memory, and learning. But here, I don't know, I always just find it very interesting that cannabinoid receptors, people take weed all the time, but cannabinoid receptors are so key for learning and memory, and maybe the drug is more dangerous than it is. Maybe it's not. That I just find it very interesting. I'm very interested in cannabinoids, so let me just move on there. Sorry. Okay. We've pretty much talked about this. This is more about the mechanism of spike timing dependent release plasticity. But this is too much detail for you, to be honest with you. But if you want to know about it, it's there, and it's there in the handout for you to work through in your own time. Okay? So, drug addiction. Drug addiction is basically mal-learning. It's learning incorrectly. Okay? So this goes for any sort of addiction, basically, whether it's rowing, smoking, cocaine, etc., etc. Right? So there's increased LTP and LTD in circuits that there's reinforcement, that's the word I was looking for, reinforcement of circuits when you take these drugs that activate reward, and like some of them are emotionally numbing, you know, so that it makes you feel better, like alcohol. So LTD and LTP are major in drug addiction. So you don't want to inhibit addiction, because unless you can target very specific addiction centers and circuits, what you're going to do is inhibit LTD and LTP. So you should never do that. It's basically arresting someone's brain development, whether they are 60 or whether they're 10. Hope they're not 10, but you get what I mean. So basically, as you can see here, the role of LTD in drug addiction is very, very key. Okay, so please don't do drugs, kids, but also a very nice point to put in your memory essay. So I've actually run very quickly because I speak very fast when I present things. But basically what we've learned today, hopefully, is that sleep and memory are global phenomenons. Okay, they involve a network of circuits, and therefore we don't actually know the mechanisms in much detail. So it involves emotions. We get very grumpy, very excited when we're very tired. It involves motor skills, as we've seen with the sports paper, cognition with Mr. Randy Gardner, and interpretation of stimuli, which you can see via the rat experiments that we didn't really go into today. In terms of memory, memory is linked into sleep and learning. LTP and LTD are hugely required, and they form a balancing act. So there's a homeostatic level between LTP and LTD that allows our brain to keep developing whilst not hitting the ceiling effect of everything's excitated, therefore nothing is excitated. Okay? And the last thing I want you to really remember is that memory is calcium dependent. There's huge amount of calcium involved in memory, and with the spike timing, it's temporally dependent. Okay, so both people in that relationship want to be in the relationship at the same or similar time.